This is the COVID-19 vaccine. It has saved an estimated 15 to 20 million people during the pandemic. And the two scientists behind this discovery have just won a Nobel Prize in medicine. Now, it has also been controversial in the last 3 years, but that's not what we're discussing today. We're talking about the science. And the science here is fascinating. These scientists had to go through a lot to get here.
Breaking news in Stockholm, Sweden. The 2023 Nobel Prize in Physiology or Medicine this year has gone to two scientists whose work played a critical role in the fight against COVID-19.
The impact of this discovery has the potential to not just prevent COVID, but also treat diseases such as cancer, HIV, malaria, and more. In the past, vaccines were produced by introducing a weakened version of the virus into the body. That's how the polio, measles, and yellow fever vaccines were made. The methods evolved to introduce viral proteins rather than the whole virus into the body. When you introduce the virus or a viral spike protein into the body, it will recognize it as foreign and start producing antibodies against it. And this is so that next time the body sees that virus, it will be ready to fight it. But producing these weakened viruses or even spike proteins in the lab takes a lot of work, especially if you want to produce them at a large scale. But as scientists learn more about how proteins are made, they started thinking about using the body to make the foreign proteins instead. And how do we do that? By giving it the foreign proteins mRNA. You see, the mRNA stands for messenger RNA. It contains the genetic information from the DNA. And this mRNA can be used as a template to make proteins. So in the 1980s, scientists found ways to make this mRNA in a test tube. It's called in vitro transcription. So if you can make the mRNA in a test tube, you can introduce it into the body and the body will start making the proteins encoded by that mRNA. You wouldn't have to make these spike proteins anymore. The body would make them from the mRNA, but that did not work. And there are a few reasons for this. First of all, mRNA is unstable, so you can't just inject it into a patient. It's gonna be degraded before it gets to the target cells. And secondly, the body reacted negatively to the mRNA. The immune system would start fighting and induce inflammatory reactions. And because of that, scientists gave up on using mRNAs as vaccines, but not Dr. Katalin Karikó. She firmly believed in the potential of mRNA for therapeutic applications. And she received a lot of backlash in her career. She was demoted a couple times. She was denied funding, denied tenure. She eventually was forced to leave her university, but she did not give up. In the 1990s, she met her research partner, Dr. Drew Weissman, at a photocopier of all places. From then on, they started working together towards therapeutic applications of mRNA. So they investigated why the immune cells attack the mRNA made in a test tube when they don't attack the mRNA inside our bodies. Well, what they found was that the mRNA inside our bodies was chemically modified. You see, mRNA is made of these 4 bases, including uridine. But every now and then, a modified uridine called pseudouridine is incorporated. So they were like, okay, if we introduce these modified bases in the test tube mRNA, is that gonna trick the body's immune system? They tried that and it worked. They showed that the delivery of this modified mRNA increased the spike protein production in cells. And also, the immune system did not react negatively to this modified mRNA. And that was a major discovery right there. Another discovery was the encapsulation of this mRNA in a lipid cage. This protects the unstable mRNA until it gets to the target cell. The original paper was written in 2005, 15 years before COVID-19 came. But these two kept working on optimizing the mRNA vaccines to reduce immune rejection and improve delivery and protein production. On May 15, 2020, in the middle of the pandemic, President Donald Trump announced Operation Warp Speed. This was a collaboration of researchers, government agencies, and pharmaceutical companies to produce a vaccine for the virus as soon as possible. And in December of the same year, the vaccine was ready for production. This wouldn't have been possible if not for the technology that these two scientists came up with 15 years before. And this mRNA technology is being used to not just make vaccines. It's being used to target cancer, HIV, malaria, and other diseases. And the way it works is the same as COVID vaccine. If you can find a protein special in a cancer, you can design an mRNA specific to that protein so that the body can start attacking that cancer. So this mRNA technology is here to stay, and I can't wait to see what researchers come up with. The possibilities here are endless. So that is the Nobel Prize in Medicine. If you want to know about the Nobel Prize in Chemistry, click the video right here, and I'll see you in the next one. Bye-bye.